the reason why I'm here is because since I was very young, I wanted to develop a drug for cancer. And I was born and raised in Spain, and I thought that if I could only do one thing in my life, I wanted to, and I had to spend it for my entire life, I would do something that would be meaningful for people. And that's what I did. And because I'm a little stubborn, even though it took a lot of effort and I had to move to the United States, I started a company to do just that, to develop a new drug for cancer patients. And when I had met in my prior years a scientist at the National Cancer Institute that had discovered a virus that would only bind to tumors and would not bind to normal cells. And that was a pretty incredible discovery, because the holy grail of cancer is we can have very toxic drugs. If we can take them where they are needed, probably we would make cancer a curable disease. So I met him and said, like, why don't we start a company together? And he said, you know, this is really exciting, we should. And we started working together. And I started the company in Boston with all of this biotech excitement and guys like Grant that put money in startups and raise capital. And all I did was raise money and more money and put it into a lab and start doing experiments. But it was a critical moment where the NCI and Aura were working, and we decided to work in ocular melanoma. And again, it's a rare disease, and why rare disease matter? And they do. For me, they matter because I have the opportunity to meet people like you. And you are the reason why we wake up every morning, because we want to develop something that helps you at the end of the day. And in rare cancers, in rare diseases, you have that incredible opportunity to be so close to patients, even from when we are working in a lab with rats and rabbits. And that doesn't happen in diseases like that have so much attention from pharma companies that put millions of dollars. And we thought that our technology was really well fitted for ocular melanoma. And when I told the NCI, why don't we start doing, and they said, you're crazy, how many patients are there? I mean, is there really something? And I said, yes, rare diseases are very important. They matter for people. And if we can prove our drug, this is a very new class of drugs, if we can prove that it works in these type of people, in these type of patients, maybe it will work for a lot more afterwards. And then we can, so I convinced everyone, and we were excited, yes, and that's where I met Grant. And we came here, and I remember the first time I presented to a community like this, we only had very little data, and I felt really bad because I don't want to overpromise. What I'm committed is to try. And so now, where are we today? So after 3 years, what we've done is we've developed the drug. We proved that in rabbits it works really well. You'll see now in a video how it works. We're trying to target the very early stage disease because we believe that if we can eliminate, if we can offer patients an opportunity to have a drug that can eliminate the tumor with preserving vision, all of these uncertain lesions or all of these small melanomas can be treated, and potentially maybe this will ultimately impact overall survival if we can catch the tumor early. If there is something that you can give to everyone that has a threat in their definition of a small melanoma, but doesn't jeopardize their vision, that would be wonderful. So that's where we're going. And so we've developed the drug. We've tested with the FDA that they are okay in going for early stage disease. We've started to talk to all of the physicians like Arun, we have Carol Shields, Evan Gragudas, all of the top physicians in the United States, and everyone is excited. We've raised $21 million, so now we have money to do the clinical trial. And we've manufactured the drug, so now we're ready. And so next year is going to be a very, very important year for all of us. And we are going to try. And the important thing is to understand what a clinical trial is. And so usually there are 3 trials to approve a drug: phase 1, phase 2, and phase 3. And phase 1, and in these kinds of rare diseases, we join phase 1 and phase 2. So that's what we're going to do the first year, which is really nice because the FDA allows us to, if it works, let's do it fast because people really need it. So the phase 1/2 is going to show the safety of the drug. We already know that it's very safe in animals. We hope that we're not so different from animals at the end of the day. So hopefully the drug will be very safe. And then we're going to test efficacy. We're going to see if the tumor shrinks or stops growing. And for these very small tumors, that's what Dr. Singh said, growth is the most important thing. So if we can stop that from growing, that would be fantastic. So that's where we are. I think that what we can say is that we're committed, we're excited, and we're not going to give up. My mother used to say, we never give up. No matter what happens, we never give up. And that's the philosophy at Aura. We don't give up, we work for you, and we're trying to make a difference. And at the end of the day, as I said, I started with a dream, and we're all dreamers. And what I know is that when a lot of people dream the same thing together, really incredible things happen. And I know that for sure. So hopefully now you'll have a little bit of a snapshot of what we've developed. It's a very small video that shows how the drug works. I think in very simple terms so that everyone can understand what we're going to try to do. And I would love to stay a little bit more to answer questions, if anything, but we'll be very deeply engaged with this community. We'll have a website, we'll know where all of the hospitals are, we'll have Allison that will be taking care of all of the patient. We want to be known for a company that's really patient-centric. I don't want to be known for a company that makes millions of dollars. I don't care. The reason why we started is to make a drug for cancer. And so that's where we want to be. And so you are a very important part of our company. And I hope that as we grow, it will stay the same. So thank you very much, and I hope that you enjoyed the little video that we have prepared.
Ocular melanoma is a dangerous, life-threatening, and often fatal cancer that occurs in the lining of the eye. There are currently no drug therapies for ocular melanoma, and so it is usually treated with a radioactive plaque placed against the exterior of the eye beside the tumor for several days. A treatment that can lead to cataracts, retinopathy, and severe loss of vision. The alternative is removal of the eye, a surgery called enucleation. At Aura Biosciences, we are developing a novel targeted therapy for ocular melanoma, an injectable nanoparticle that can kill and eliminate the tumor in an outpatient treatment setting. The nanoparticle binds selectively to tumor cells and their surrounding matrix. Infrared laser light causes the nanoparticle to release molecules that destroy the tumor cells, leaving healthy cells untouched. The nanoparticle is the outer capsid of a virus, without any internal machinery. This viral nanoparticle is a protein scaffold that is incapable of reproducing. On the viral nanoparticle surface, we attach a fluorescent molecule that is non-toxic. The viral nanoparticle is administered by an ocular oncologist by first injecting into the eye and then, after a few hours, shining a beam of infrared light to the tumor with a laser. Under infrared laser, the fluorescent molecules bound to the viral nanoparticle are activated and disrupt the membranes of tumor cells, destroying them completely. Aura Biosciences, working to provide a treatment for ocular melanoma without surgery or radiation, using targeted therapy in an outpatient procedure.
Answer any questions that you may have.
Do you envision this drug being able to attack the cancer cells circulating in the blood, or is it just attack only the tumor?
So at this moment, the drug, the viral particle just attaches to the tumor, but the drug needs to be activated with infrared laser to be very, very potent. And that's what kills the tumor in the eye. That's our first drug. Once we show that we can kill tumors so selectively, we could potentially develop a systemic drug that would work very similar. If we can direct drugs to the tumor, then we could direct them to the liver too. But then we have to use another drug that it's not light activated to be accessible. And we're working with that. So once, we're a very small company, so the first thing is to make sure that the clinical trial shows efficacy And that will open up the opportunity there to hopefully raise more capital and do other things. We're very excited. The animal models show that it binds very selectively to tumors, even outside the eye. When we put the tumor in the flank of an animal, it will go there. So yes, it can be very promising, and we're committed to the disease. Because this is a rare disease, and we want to make sure that we can provide something for the primary, and in the future, something for the metastatic as well.
Hi, over here.
Yeah.
So as I was watching the video, and how it kind of exploded the tumor, thanks. A vision popped in my head of all those cancer cells spreading
Yes.
throughout the rest of your body. So is that part of the trial that you're going to be watching? Or?
Yeah, so the most important thing is that the cells die immediately after the drug is activated. So whatever is eliminated, there is part of cell debris that's dead. So there is no spreading or bombarding of tumor cells everywhere. It's whatever, the video sometimes shows things that, what we see is that the cells die. And they're basically very similar to radioactivity probably, that we don't know whether they will just be eliminated gradually, all that cell debris that's dead, basically, whether it would leave a scar of tissue. Sometimes we've seen that when I've been talking to physicians sometimes. So we don't know whether it will stay the same, whether it will reduce. But there's not a spreading of, at least we haven't seen that in any of the models that we've seen.
So you've done the testing in the actual animal's eye?
Yes.
Oh, okay.
So the way we do this is actually, we've worked with two very good ocular pathologists, one at Emory University and the other at McGill. And what they've done is developed a rabbit model where they inject ocular melanoma cells from humans in the choroid. And after 3 weeks, these are extremely aggressive tumors, nothing like what would happen in a human, but it allows us to evaluate it in a short period of time. So after 3 weeks, we have already melanomas that are 3-5 millimeters, and if we leave them another 2 weeks, they will be 10-15 millimeters. So they grow exactly under the retina and in a very similar structure and form. So we're able to evaluate what's probably as close as we can to the phase 1 clinical trial in a patient. And so what we do in these animals, we give an intravitreal injection of our drug, and then we activate it with the laser as well. So we set up nearly the same as, not as fancy as the Cleveland Clinic, but we do it in a very similar fashion to make sure that the drug works. And what we've seen is that after 3 injections, so we do 1 injection, and then after 1 hour laser, then the following week 1 injection, the tumors disappear. And then we do histology. So in this case, we were able to remove the eyes of the animals. We cut them to make sure that there's any tumor cell left. In some cases, if the tumors, we go very big, so even like 15 millimeters. In the rabbit eye is half of the rabbit eye. We see that the tumor is still there, but it's completely dead inside. There's no tumor cell left. And if the tumor is small, it disappears. So, and the retina doesn't seem to be damaged. So we are hopeful that this is what we will see in the clinic. We're going to start with the small tumors to start with. The FDA is very supportive of early stage treatment, because in this disease, what we think is that we have the beautiful opportunity to treat early. If we have the possibility, of course, without damaging vision, if we have something that's non-invasive, very targeted, we should be able to treat as early as we can. That's the goal of cancer. Prevent or treat early, because that we know that the earliest you treat, the metastatic rate decreases. The chances of that cell escaping is less. So that's where the FDA wants us to go, which is very unique, because in cancer, usually they don't allow you to treat so early unless it's something as obvious as this one. And we're very happy. We were very nervous. We would not have been able to convince an investor like Grant. It's not Grant's VC, but investors are very, very big, and they wanted the confirmation that the FDA would allow us to test in this kind of primary setting. And we feel very, very excited about it. So let's see that humans behave like rabbits, and then we're all set.
One more question.
I was wondering if you could use this technology to apply to ovarian cancer, breast cancer,
Yes.
et cetera.
Yes.
And you think it would be just as effective?
We have animal models in ovarian cancer, in head and neck cancer, in lung cancer. We've shown that the virus is very selective to all of these cancers as well. So the opportunity obviously is after ocular melanoma, we could potentially do other cancers. If the selectivity is what we see in patients, then it can be a very nice new class of drugs. Yeah.
Thank you.
Sorry, I mixed one more. Yes. Big mouth. I'm just wondering, other than my love of carrots, how did we get into this clinical trial? I'm a rabbit lover, but I want in on this.
We have a very nice clinical team at Aura that we've built this year, and we're working with the top ocular oncologists in this country, including Dr. Singh. So we don't have now the list of hospitals. It will be available very shortly. And the process is the following. We will have a website that will use the newsletter of the OMF to make sure that the information is available for everyone. And there will be a specific criteria to get into the clinical trial. That's usually how clinical trials are done, so that we can have patients that don't have other diseases, for example, or that have other complications in the eye that may, that make the interpretation of the results not so clear. But it will be very, very shortly. We are now submitting the final package to the FDA, and so the FDA needs to review all of our data carefully. We hope that by January we'll have the okay from the FDA, and then we're ready to go. Then the 10 hospitals will be initiated and we'll start enrolling patients.
That's great. Thank you, Ellie.
Thank you so much.